Hello and welcome to our second Death and Decomp podcast. My name is Dr. Amanda Rowe.
And I'm Dr. Leon Higley with my beverage of choice today, which is ice water.
Oh, my beverage of choice is vanilla rooibos in this beautiful mug that I picked up in Lindsborg, Kansas at a cute little cafe.
What? You're drinking what? Is that tea?
It is tea, yeah. Vanilla rooibos. It's the red tea. It's a red tea.
Do you know why there's a thumbs up sign on the screen?
I don't, but I like it.
I didn't put it there. I feel like somebody's invading our privacy and posting things. As you can tell, we're not in the same geographic location. I'm sitting in my dining room, which I've turned into my office, much to Phyllis's chagrin. And look at you, all next to the Christmas tree.
Yeah. I'm recording from my humble abode on this beautiful first major snow of the year. Major's giving it a little too much oomph, but first snow. So
yeah, no snow down here in Lincoln.
What?
Yeah, nothing.
Oh, it's snowing here. It's great.
So because you're not here, you're not getting the Christmas gifts I had for you, which are finger puppet.
Yes.
Vultures.
It's my favorite bird. Well, second favorite bird.
So they got different color heads. So I'm going to say one's turkey and the other's a black vulture.
Okay.
Yes. And
nice.
Then someplace around here, I've got some really cool ants that I also bought for you. So
thank you.
Suitable for Christmas decoration. So I should have sent them in. I should have sent them in with Phyllis this morning if I'd known we weren't going to get together.
I don't have any cool things to show.
So we're trying hopefully some improved sound and graphics. And I've got some lighting to try to make my pale face wash out even more significantly. I actually could have changed the temperature on that light and made it warmer, but I'm just going to go
next time.
Yeah, maybe not. So how do you want to start? Should we talk about, I guess we can talk about gossipy things to begin with.
Yeah, let's gossip first, because I have a great story to tell you that I told Phyllis. Hopefully she didn't tell you already.
No, go ahead.
Okay. So a couple weeks ago, I had got some maggots for lab research, and I think we talked on the last podcast about using them. You and John used them for some heat shock protein work. And I had left them in a container in my office with the lid on. And when I was there yesterday, one had emerged, and I was like, okay, great. I'll let a few more emerge, and then I'll go put them in their cage. I was running late today because the roads were slippery and traffic was going very, very slowly. And so I ran straight into the office. I printed some final exams off, and then I actually went to my physical office. I opened the door, and it was like the apocalypse. The lid had come off of that container, and I had hundreds of giant blue bottle flies loose in my office, flying all over the place. And I wasn't expecting it. So I just opened the door and I looked, and they're swarming my face. It was out of somebody's probably worst nightmare. Not mine, but other people's maybe. And I just was like, I don't have time for this. And I closed the door and I went down and gave my final because I was already running a couple minutes late. A couple of them got out of my office, and I was like, oh my gosh, this is it. I'm gonna get an angry email from someone because there's flies loose in the hallway. And normally they're in the lab, and I have a seal under my door, so anything that gets out stays in the lab. It failed. Anyway, it failed. So they were all over. After the final, I went back up to my office. I found my fly swatter. I killed as much of them as I could, as many of them as I could. Was not very efficient. So then I just got my net out, and I was sweep netting all through my office, trying to get all of these flies that were up on the ceiling that were too tall for me to get to fly swatter. And then I went out and I killed a couple in the hallway. And then I had to let the custodial person know, hey, just so you know, there's nothing dead in the building. Some flies escaped. I killed as many as I could. Oh my gosh, it was a whole thing.
So did you took the flies you got in the net? Did you put them in the colony?
No, flies out of the cage are dead flies. Lab rule.
So you're selecting against escapees. Is
Yep.
that the idea?
Yep.
That somehow that sounds wrong to me.
I want them sweet and docile and homebodies.
So did you put the container then in a
I did.
cage?
Yeah. Yeah. There were still quite a few in the container. So
I just like to say this is a perfect example when you should have had the bug assault gun that you didn't let me use in the lab.
the bug assault gun and the slightly electrified tennis racket.
That's right.
Also, yeah, also
I
would
still have a bug assault in the garage, but I don't know where the electrified tennis racket is. But that we definitely that should be a requirement for your colony work.
it should be because I broke my fly swatter and I had to use lab tape to tape it back together, but I think it's done for now.
So
It
if
was
you
not
don't know
made
about
anymore.
if you guys don't know what bug assault is, it's a really cool looks kind of like a water gun
Like
that
a Nerf
shoots
gun, yeah, like a
or a
yellow.
Nerf gun, and
Yeah.
it shoots little puffs of salt that will kill a fly if you get it close enough and have a good enough aim. I would like to point out that I was like a quadruple ace. I think I'd shot 13 or 14 escapees in the lab until Amanda said, in no uncertain terms, I was to stop it because she was sick of me getting salt all over the lab, if I remember correctly.
I
So,
think for a while we were even keeping tally on a
oh
whiteboard.
yeah,
There was
a
a
tally.
kill list.
Yeah, we
Yeah.
had a tally board.
Yeah.
So, yeah, I was
So
feeling
that's yeah,
pretty proud.
that's
So
my news story.
that's awesome.
Yeah. Largest
I don't know if
jailbreak
it's
I've ever had.
I don't know if it's in the gossip side, but I finally decided I should put in my membership. I put in my application to become a full member of the American Academy of Forensic Science after 15 years of being an associate member.
Yep.
So I did that this morning. And in the process of fiddling around with that, I came across a couple of papers, and I don't know if you'd seen these, but I thought they were worth mentioning on the podcast. The first was a paper by Jeff Wells, which is really good. It was Jeff, I want to say settling the hash. Is that still a phrase? But putting those jerks who said that you can only do PMI minimums in their place. So he has a paper that shows that not only is that not true, but the people who purported it, who claimed it was true, turned right around and in testimony violated their own rules. And so he has that case study from Las Vegas. It's that case study where there was a body by a dumpster. It was a murder, and there were no flies on the body. Do you remember this case?
I vaguely remember this case.
So the gist of it was there were a whole bunch of experts for the defense who claimed that the flies definitely had to lay eggs in the 2-hour window, and so before the body was found, before dusk. And Jeff, who was a prosecution expert, said that's absurd. You could have lots of circumstances where flies might not find a body within 2 hours. Mm-hmm. But the fact that they were adamant about that shows that they were adamant about using successional data to show that, successional data. We should explain. There's a sequence of events.
This is a vocab for villains.
Okay, vocab for villains. Vocab for villains. So succession is an ecological phenomenon where a resource changes, and then the organisms that are associated with that resource also change. And the classic example is like a new island that arises out of the ocean, and you have what are called pioneer species. And then they help create soil, then you get different plants, and it progresses. Well, the same is true with dead bodies. And in fact, here in a minute, we're going to talk about that. We realized that as the Death and Decomp podcast, we should really spend some time actually talking about decomposition.
Yeah.
So, the other thing that I found that's in the same vein that was interesting was a paper that's just published, which claims that you can use bacterial contents of the appendix to determine the postmortem interval. And based upon studies they did using genomics and rat appendices.
Okay.
Which sounds like an interesting idea, and they have nice graphs, and they have lots and lots of charts. But Amanda, there was a tiny flaw I detected in the paper. Would you like to guess what the flaw is?
Well, I haven't read the paper.
I know.
So. I don't know. You can just tell
me.
Me.
Replication.
Oh, well, okay.
So their experimental design, they used a whole bunch of rats, but it appears to me that when they actually did their data, they pooled everything. So they had replicates going in, but looking at their charts and so forth, I don't think they used them because they don't show measures of variation. And in particular, they had a regression line, and they should have had stacked points on the line if they had regression. They didn't. The points were along. So I'm pretty sure, I mean, I can't be certain because they weren't explicit in the writing, but I'm suspicious, which we'll return to this topic over and over again. But for those of you who aren't scientists, when you do an experiment, one of the requirements for an experiment is that whatever you're testing must be repeated. And each time you repeat it, that's called a replicate. And if you don't repeat it at least one time, you don't have an experiment. And that's because you can't distinguish between what you think is going on versus whatever might be randomly occurring. It has to be repeatable. Generally speaking, a magic number is usually, we use 4 replications, but in some studies you might use 12. Rarely do you use fewer than 4. That really only happens when it's very, very hard to do the experiment. So we'll have more to say about what the key components are of experiments, but I thought it was interesting, the idea.
Yeah.
I mean, it sounds sketchy to me. Even if it worked one time, it seems unlikely to me that it would work every time. I'm assuming even with rats, which were their experimental animals, that the gut flora and fauna are dependent upon diet and some independent genetic variation.
Right. And those, I mean, rats in colony are probably fed the same things. So bacteria.
And nobody's going to spend the money to be doing tests on appendices. Can you imagine trying to do that for how large a data set you would need to actually prove it for human beings?
Yeah. Also, as someone that is appendix-less, you know, that's not very helpful.
Yeah. So that falls into the category of what I would call peculiar ways people keep trying to estimate the post-mortem interval.
Most common one, I think, probably, at least when people talk to me, is using vitreous humor, the fluid in the eye.
You mean the long proven to be incorrect vitreous humor method? What, Amanda? What do they actually?
There's still people that are like, we can use it, and I'm like, I don't think.
What did they actually measure in the vitreous humor? Do you know?
I think potassium.
So the idea is there's decline, or which way does it go? A decline or an increase?
Unknown. I don't know.
I guess we should look that up. Yeah, so we'll get back to you on that. The important thing is it doesn't work.
It doesn't work. So it doesn't actually.
Other than that minor point, there's a perfect way to do it, other than the fact that it doesn't work. So.
Yeah. Also, plus, like, by the time we usually get called, there's no eyeballs left. Not very often. And
also a minor problem, right? No, the lack of eyeballs, that will do it.
Yeah, there's, you know, sometimes there's no soft tissue left by the time we get called. So things like that. I mean, I guess if you're looking for an hour count within the first 24 hours, maybe. I mean, it doesn't work, but if that's what your time frame is, you know, there might be some sort of biological thing like that you can use. But yeah, by the time we get called it.
Okay, Amanda, do you want to, we said we'd talk a little bit about decomp. Can we start talking about the so-called, except they don't really exist, stages of decomposition?
Yeah, let's talk about them.
So I'm happy to do illustrations. I'm in the midst of writing a paper called "The Value of Bad Drawing in Teaching." So
do we want to like switch off? Like, I'll start talking about fresh, and then you talk about the next stage. And
So this guy suddenly realizes he's about to be a subject of decomposition, so he's not looking too happy. All right.
Yep. Okay.
So fresh, Amanda.
So let's start with our first stage, and I'm gonna put stage in quotations, because when we talk about the stages, of which I use 5 stages, some people use 4, some people use 6, I think. When we talk about the 5 stages of decomposition, remember that they're not discrete stages, so you can have overlap between the different stages. So on some bodies, you'll have some bloat, some discoloration, some active decay, some advanced decay. So we can like sort of mix stages together. So we're going to talk about them as separate stages, but just remember that they are not discrete. They're not separate stages where one ends and the other one starts. So we are going to start with fresh stage. As the name implies, this is a freshly dead body. As you can see, the heart has stopped pumping blood, the brain activity has ceased, and what we see happen during the fresh stage is autolysis, auto meaning self and lysis meaning to break. So we start.
Oh, another good word. We get a double vocabulary.
Yep, vocab for villain, autolysis. The cells start to rupture and break open.
And it's in the fresh stage that we see the rigors, right? So we have rigor mortis, which most people know. And you want to explain how rigor mortis works, Amanda?
So rigor mortis is the contracting of skeletal muscles of the body, and it can occur anywhere between 0 to 48 hours, I think. It depends. It's really dependent on muscle mass and fat mass. And so some people never undergo rigor. Some people undergo rigor and it lasts for a long time.
And so the story I tell, my former student, Tim Huntington, worked for a mortuary in college, and he did a pickup of, he told me, a former Mr. Universe, and they were at the house 20 minutes after death. Apparently there'd been sort of a death watch, and in the 20 to 30 minutes it took them to get there, the individual who's still in phenomenal musculature had already rigored up to the point that they kind of had to carry him like a board out of the apartment. And so if you look at an overweight guy like me, I might never rigor up. So it's very much tied to fitness, and it's not permanent. Rigor sets and then it will release. And the pattern of rigor,
as the proteins in the muscles start to break down. Yeah.
It's also different with children. And so the onset is a little bit faster, and the if I remember correctly, and the dissipation shorter. It's a shorter time interval with the young. So what other things are associated with the fresh stage, Amanda?
Skin color looks about normal. They might be a little bit pale, but
we mentioned rigor mortis. The other thing, the pale skin, that's the other livor mortis, which is
the settling of blood or pooling of blood due to gravity. So we see blood start to move down and settle depending on how the body's laying.
And that turns out to be important because that will set in place. And if the body's moved, it will still stay the same. So you can sometimes look at where the blood's pooled, and if it's not at the lowest points, you'll know that someone moved the body after livor mortis had set. And so again, that's, I don't know, a matter of a couple hours. It starts settling right after death, just as soon as the heart starts pumping.
Yep, as the heart stops pumping, the blood, there's nothing moving it, so it just follows gravity.
Now, the interesting thing from Amanda and my standpoint has to do with these guys.
Yep.
So how long does it take for insects to come to a dead body?
Not very. So we use pigs in most of our research because it's easy to get replications. We mentioned that earlier in the podcast. You can get the number of pigs that you need to make sure that what you are seeing is true and not just some one-off weird thing. We use pigs, and I have been with the pig. So the pig dies, we carry them to the car or to the back of the truck, and there's already flies on them. So 30 seconds about, which to me is fascinating. So I teach a lot of anatomy and phys, human anatomy and phys. And from a human standpoint, we still argue about what death means. Like, when does death start for a human being? Is it when the heart stops? Is it when the brain stops functioning? And so we're arguing about that, where insects have evolved so well to finding resources that they can tell immediately with some sort of chemical change that happens that these tissues are now decomposing and they are ripe for egg laying, which is just, it's like mind blowing.
It is mind blowing. And I always, I joke, and it's not a joke about this, that there's really only two alternatives to explain what's going on. And neither one, both are disturbing in their own ways. The theological argument is that flies can detect the departure of the human soul, which I don't even know what that would mean. And as a guy who doesn't believe in the existence of the soul, it's kind of problematic for me. So that's problem number one. An alternative scientific explanation is that there are decompositional chemicals that suddenly appear, and by suddenly I mean within seconds after death, certainly within 30 seconds, so the flies can detect these. Presumably those decompositional chemicals are from bacteria. Most of the things that we associate with really bad smells are sulfur-containing compounds. There's a class of compounds called mercaptans that are particularly nasty. And it would have to mean that your body is keeping those microbes at bay until the instant of death, at which case the things that want to eat you start eating you. And both explanations are kind of alarming in their own special ways.
I think it makes you really appreciate your immune system just a little bit more.
I'd appreciate it more if I didn't have this cold and it had done a better job for me. But
yeah,
this
is actually a really good segue into our second stage of decomp, where we really see autolysis take off. Right, cells are breaking open, bacteria are flourishing. So, Leon, lead us into our second stage with your beautiful illustrations.
I'm working on it. Well, okay. I'm going to go with this. This may not actually be accurate, but close enough.
No, I think that's an accurate representation. Although you need the arms and the legs to be sticking up too.
Oh, okay. This is one possibility. Let me draw you what I think the other possibility is.
Okay, well, he's drawing. Well, I'll jump right in. So the second stage of decomp is called bloat, and this is caused by the stopping of the immune system, keeping those bacteria and those other microbes in check. And so the bacteria that are found in the gut, excellent.
The head has been defleshed by the maggots.
I like it. I like it.
Okay.
So the gut bacteria that were kept in check by the immune system now just explode, and one of their byproducts is gas, and that gas builds up in the body and it leads to bloat. And everybody's probably seen bloat. If you've ever seen a dead deer or a dead raccoon on the side of the road in the middle of the summer and its legs are sticking straight up and it looks like it's a balloon, that's bloat.
So an interesting story about bloat is that people always want to ask me what happens at the end of the bloat phase. And I had a pig on campus. Amanda's, I know you've heard this story. I had a pig on campus that I'd taken a class to, and it was in full bloat. And I had had students coming around, and they were scared, but as they stopped getting this, you know how it is. Yep. They realized it didn't smell as bad as they thought it would. They were getting close. One student says, "Now, what happens at the end of the bloat phase? Does it pop?" And they kicked it. And what did we learn? We learned one, it does pop.
Yep.
And two, you're never getting that stink juice off those tennis shoes. Those go straight to the trash pile because, oh my God.
Yeah, so, it's the smell it keeps on giving for sure.
Yeah, so it's an interesting theme that we'll have a theme of bad smells. Last week was adipocere. This week is the juices associated with decomp. And I think there's a significant amount of methane that's typically in there as well. And I say that because of reading a story of someone who lit a bloated cow and had it explode. So no matches near bloated animals. That's a good piece of advice.
I think they blew up a whale on purpose, right? But they used dynamite for that. I don't think it had to do with methane.
Oh, yeah, that's a wonderful. I'll put the link So let me write the note. I'll put the link in. That's a classic YouTube video when they could have killed, I don't know how many people by blowing up the whale. The proper disposal of whale carcasses is to haul them out to sea. And I know that definitively because of Marcus the Carcass. One vacation that we were on, there was a whale that washed up on the beach, which we promptly named Marcus. And I was having great fun watching decompose on shore until the authorities decided they should take it out. And I learned that they present really great successional islands on the seafloor.
They do.
There's a massive array of organisms that feed because there's a sudden influx of these nutrients in what's otherwise a relatively nutrient poor surface.
Yeah,
well,
they have some really great. They have some really great videos. Okay, so along with the extension of the skin of the body during bloat, we also start to see some skin discoloration. So it goes from normal looking skin, you start to see some marbling, which is just introducing colors like green and purple into those tissues. And you might start to see some skin slippage, which is where the epidermis starts to separate. The epidermis is the top layer of skin where it starts to separate from the dermis, which is the bottom layer of skin, and literally slips off the body. So you'll start to see some of that happen if bloat is really extending the body a lot. So unfortunately for those bacteria that are causing that buildup of gas in the body, they are anaerobic, which means they dwell and thrive in areas that have little or no oxygen. So the minute you introduce oxygen into the body, most of those microbes die off. So when the body pops from students kicking it, or vultures poking holes in the abdomen, or just it pops like a balloon on its own, there they leave these nice little head-shaped holes right in the abdomen as they poke through.
Or the skin just lets loose.
Or the skin just, yep, just like a balloon. Excuse me. Air rushes in, kills off most of those microbes, and then we are really just heading fast like a train into active decay. So in active decay, let's see your representation. I need lots and lots of maggots. Oh, come on. More maggots than that.
I can't. That's just guts. I can't really draw maggots to scale.
Draw them like little lines. Just cover them in little hatch marks. So in active decay is where we tend to see a lot of insect activity, with the eggs having been laid previously during. Very good.
Thank you.
During fresh and or bloat stage, or even sometimes during active decay, you'll still see eggs being laid. So maggots have really started to take off. They're really going through and consuming a bunch of that soft tissue. So things like muscles and skin. Oftentimes the head by now is mostly skeletonized, and then they start moving down into the abdomen.
Yeah, sometimes you can get a skeletonized head. Yeah, I guess you're right. Usually it's post-bloat. It doesn't take maggots very long to skeletonize a head. And when they skeletonize it, they don't just take the skin off, but they'll remove the contents of the brain as well.
And the eyeballs.
And the eyeballs. And we should mention why they go for the head. It's that they have crap little mouth hooks.
Adorable little mouth hooks, you mean?
Yeah, but
attached to scientific term called the cephalopharyngeal skeleton.
I love that phrase.
Yep, down. There we go. Down. Down. There. Oh, there's the butt. Up. Up. Okay, stop. No, it's not focusing automatically. Pull it back towards you. Okay, there we go. Okay, stop. That's good. So the pointy end that you can see, that's the head end. Those are the mouth hooks. The broader end where it looks like two eyes, those are actually not eyes. Those are the maggot bottoms, and those are their posterior spirochelar plates. That's what they actually use to breathe while their heads are down in gooping, decomposing tissue.
And just like that Christmas favorite, keeping with the season, Die Hard, which has the famous...
That is one of my favorite Christmas movies.
The famous ductwork scene. Insects breathe through ductwork. They don't have oxygenated blood, but that's a whole thing.
Yeah.
So I'm a guy who only has one more stage. What stages do you have?
So advanced decay and skeletonization. We
Really?
can combine them. Yeah, we can combine them.
And I just say remains, but
Okay.
you're okay.
So advanced decay is where we see most of the body mass being consumed and gone. We see the maggots move away from the body,
Oh,
and
let me.
then we enter into the remain stage where it's mostly bone. You can see some bits of skin. There might be some leatherized pieces of skin. Oh, there we see migration. So those maggots are moving away, finding a nice place to pupate and form their little fly cocoons so they can emerge as big, beautiful flies, like the ones that were in my office today.
So some people will call the remains the Indiana Jones stage.
Oh, yeah.
I don't know if you've heard that before, but typically there's no soft tissue left, and it doesn't mean there aren't insects. There are some beetles that
Beetles
feed
mostly.
on dried skin, and beetles that feed, and there's mites, not an insect, but still an arthropod.
Mm-hmm.
So
Yeah. So, did we miss anything major? I don't think so.
no, I don't think so. There's a lot more that can be said about this, and we probably will.
If we don't have, oh, let me just say, if we don't have insects doing most of the removal of biomass, we see that with vertebrate scavengers. So wild dogs, birds, wolves, coyotes.
Skunks, raccoons
Skunks.
occasionally.
Yep. I've seen box turtles. Strange
Wow.
box tortoises. Yeah, strangely
Somehow
enough.
that seems very wrong.
It was jarring. Yeah.
Yeah. Um, so so you have to realize the body is just essentially food. So it's
Mhm.
things that can come and feed on it. And Amanda mentioned that post bloat you have this oxygen. Oxygen plays a really important role in decomposition. The bacteria that are responsible for decomposition, some live with oxygen, they're the aerobes, and some without, the anaerobes. And the anaerobes are really slow. And
Mhm.
so the more oxygen that gets to the tissues, the faster the bacterial decomposition. But truthfully, the loss of soft tissue is really accelerated by vertebrate scavengers and by insects.
Mhm.
They open the body up, so it'll also speed up bacterial, but you can have very, very rapid loss of tissue through those agencies. So let's talk about timeframes, shall we? How long, Amanda, does it take to go from fresh to remains?
Well, if we're talking about insects doing most of the work, it's temperature dependent. If we're talking about vertebrate scavengers doing most of the work, it could go very quickly and we could skip some of those other stages. So we can go from fresh to remains in a couple of hours if you're on the African savanna, and a couple of days if you're in the woods of Colorado, for example, and an elk dies, where they just will denude the bones completely of all soft tissue just very, very quickly. So
another thing you need to be aware of is we presented those stages, but you can look at it's not even across the body. You can look at a hand, for instance, and you might say, oh, this hand is barely decomposed. And yet you might see that the head is skeletonized and you're in a post-bloat stage.
yep.
And so the rate at which different parts of the body decompose are a function of oxygen availability and what's feeding on it. And so those parts of the body, feet are a good example, that tend not to decompose at the same rapid rate unless they're in a fluid or they're somehow made attractive to insects. And so lots of people have tried to do numerical schemes for rating decomposition, and you can find papers going all the way back to the 1800s with 7 or 8 or 16 different stages. But the reality is far, far more fluid, and almost all of this is temperature dependent. And so with insects, we could look at going to the remain states in maybe 10 days in the summer
in the summer. Yeah.
But by the same token, under spring or fall temperatures, we may see that same process take a month. And so that's a lot of where Amanda and I try to use our expertise when we're evaluating times of death.
Yeah.
Is
looking
some
of it, some of it's moisture related too. So bodies will decompose differently in the desert, for example, than in the Pacific Northwest. And so
and may not decompose, may dehydrate and mummify.
Yeah. So again, these stages are not discrete stages, and they will not be the same for everybody that is breaking down.
And as a rule of thumb, you can use as a guide that whenever anybody tells you they know what the story is, they have it's just like Amanda and I joking about a new method to figure out the postmortem interval. Lots and lots and lots of efforts are made, and every year we find new papers. But the reality is it's a complex problem, and I very much doubt anybody's going to come up with a simple formula that's ever going to establish this, because it's a complex biological, biochemical, and even physical process. When you look at the remain stage, bones are weathered, and bones are affected by rainfall and freezing and a whole variety of those things. So one of the ironies is sometimes if we're looking at a remain stage case, we may be able to tell you the season when the victim died, but we can't tell you the year.
Nope.
Which seems kind of ironic. So we'll have more.
Cutting it down to 4 months and no year is better than 12 months and no year, right?
Exactly right. Exactly right.
Yeah.
So let's see. We talked a little bit about beetles. I suggested we use that as our bug of the week. So let me get a picture. I'll be right back.
This week's bug of the week is brought to you by Leon. I'll do next week.
So this is a takeoff of a famous, it's a small rendition of a famous painting by the German artist Albrecht Dürer. And this is Lucanus cervus, the European stag beetle. And stag beetles are cool. The immatures tend to live in dead wood. You can rear them on, I think they'll eat apples, which I never understood why you can feed stag beetle larvae apples or dried fruit if in the real world they're feeding on decaying wood.
Yeah.
And they're big. This beetle is in life quite a large beetle, something like this. And you'll find it on stamps. Here, I'll put this up while I'm talking. You'll find it on stamps, and it features in a lot of art. This is a male beetle. The size of those mandibles are used by males to fight for females. They'll meet each other on a branch, and then they use them like levers to try to knock the other tiger beetle or the other stag beetle off. And I told Amanda before we started recording that this was my choice for a Christmas insect for a very bizarre reason. And I don't pretend to be the author of this. This is just what I read. What I read is that this species of beetle was associated with the god of thunder, Thor.
Oh.
Because lightning-struck trees, which would die, would frequently have stag beetles come out of them. That makes sense, right?
Yep.
What doesn't make sense is that after the Catholic conversion, to Christianity, this beetle became associated with Jesus Christ as a sign of rebirth.
Oh, resurrection.
Resurrection.
Okay.
And I can only assume it has to do with life coming out of death, as in dead trees.
Yeah.
But I'll tell you, it's a stretch. I'm willing to believe it's true, because why, for example, did Albrecht Dürer, most of whose subjects were religious subjects, not all of them, but why did he choose this of the only insect he ever illustrated?
Yeah, interesting.
So
Happy holidays.
Happy holidays, thanks to the stag beetles.
That's very good.
Yeah. So I think is that it for this week? We wanted to be a little bit short.
My cup's getting low.
And my cup's low too. Okay. Well, look, I hope everybody has happy holidays. We'll be back right after Christmas with episode number 3. And I can't tell you what that will be, but I'm sure it will be just as peculiar and potentially disgusting as all of our other episodes. And
I'm really looking forward to some more illustrations.
Oh, well, I'll get to work on that.
Okay.
Okay. Thanks.
All right.
Bye, everybody.
Bye, everyone.
Hit like and subscribe.
Hit like and
That's right.
subscribe.
Okay. Bye.
Okay, bye.